Hello everyone and welcome to Prof Talks. Today we will be discussing a very important ICH guideline, that is ICH Q3D on elemental impurities. This guideline have been published in March 2020 and this is the first revision of this guideline. So let us review this guidance. If we look at the elemental impurity guidelines, there are four important guidelines, the first of which is ICH Q3D revision 1, which was issued in March 2020. Second is the USFDA guidance on elemental impurities in drug products of August 2018. There is also a US Pharmacopoeia Chapter 232 for elemental impurities limits, which was published in August 2017. And US Pharmacopoeia Chapter 233 for elemental impurities procedures of August 2017. It should be noted that US Pharmacopoeia has removed the Chapter 231 on introduction of the Chapter 232 and 233. The Chapter 231 was limited to the reduction of heavy metals. So in this presentation we will see what part, that is what are the elemental impurities, how are they classified, what is the maximum allowed limits of these elemental impurities in the pharmaceutical products. From which part we will see to which product this guidance is applicable to. From where we will see that from where these impurities can be introduced into the product. And the how part we will see how to control these elemental impurities. Before moving ahead, please ensure that you have subscribed to the channel Prof Talks and please press the bell icon so that you receive the notifications of the upcoming presentations. Starting with what are elemental impurities? Elements in a product which possesses toxicological concern and do not provide any therapeutic benefit to the patient are called as the elemental impurities. They can be catalysts or environmental contaminants that may be present in drug substance, excipients or drug products. They can occur naturally in a product, can be added intentionally during the processing or introduced inadvertently, that is by interaction with the processing equipment, container closure system, etc. So there are 24 such elements. These are considered in these guidances. All the guidances, that is ICH, USFDA and USP Chapter, aims at setting an acceptable limit of elemental impurities in the drug products, update the methodology used for testing of elemental impurities in the product. If we see the scope of this guidance is applicable to all the finished drug products, specifically which include the purified proteins and polypeptides, their derivatives, also to synthetically produced polypeptides, polynucleotides and oligosaccharides. This guidance does not apply to the herbal products, radiopharmaceuticals, vaccines, DNA products, whole blood components, plasma derivatives, product based on genes, tissues, cells and clinical research products. So if we see the classification of these elemental impurities, the 24 elements have been categorized in three categories: type 1, type 2 and type 3. Type 2 has two subcategories, 2A and 2B. So the categorization has been carried out based on likelihood of occurrence of these elemental impurities in a drug product, that is probability of use in the process, or observed natural abundance and environmental distribution. Also, toxicity of the elemental impurities has been considered for the categorization. Toxicity is based mainly on the permitted daily exposure, that is PDE value. The details of PDE we will see in the forthcoming slide. So for type 1 impurities, these are limited and there is no use in the drug products. They should be evaluated and thoroughly evaluated into the drug products. They include arsenic, cadmium, mercury and lead. Type 2, that they have a high probability of occurrence and should be considered for evaluation into the drug product. They include cobalt, nickel, vanadium. Then there is type 2B. They have a reduced probability of occurrence and it can be excluded from evaluation because they are not much toxic and do not found abundantly in nature. So these include 10 such elements, that is silver, gold, iridium, osmium, palladium, platinum, rhodium, ruthenium, selenium and thallium. And finally the type 3. They are low toxicity through oral route and can be excluded for oral medication, but need to be considered for inhalation or parenteral route. They include barium, chromium, copper, lithium, molybdenum, antimony, tin. So we have seen that toxicity of these elements is evaluated through permitted daily exposure, which is termed as PDE. We will see what does it mean. PDE is the maximum permitted quantity of each elemental impurity that can be contained in maximum daily intake of a drug product. It is expressed in microgram per day. So the consideration of PDE is mainly based on three routes of administration, that is oral route, parenteral route and inhalation. If we see the calculation of PDE, it is mainly considering some aspects and we will look what it is. First is NOEL, that is no observed effect level, that is high dose at which there are no biologically significant increases in frequency or severity of any effect in the exposed organism. Second is NOAEL, that is no observed adverse effect level, that is greatest concentration of a substance, that is dose, that causes no adverse effect on an organism under defined condition of exposure. And third is LOAEL, that is lowest observed effect level, that is the lowest concentration of the dose that causes adverse effect on an organism distinguished from normal organism under defined conditions of exposure. If we see the formula for PDE, it includes NOEL mainly, and if it is not available, NOAEL into mass adjustment by 5 factors, that is F1, F2, F3, F4 and F5, where F1 is the factor for extrapolation between species, F2 is a factor of 10 for variability, F3 is variable for toxicity studies for short-term exposure, F4 is factor to be applied for severe toxicity, and F5 is variable if NaOL is not established. So the thing is, seeing at the complex formula, we don't have to worry about the calculations because the guidance have already provided the permitted daily exposure limit for all the 24 elements for class 1, class 2A, class 2B and class 3. So if you can see into the table, the oral PDE microgram per day, parenteral PDE microgram per day and inhalation PDE microgram per day have been clearly defined, and every product should comply to these requirements. Now we move ahead through the compliance to the ICH Q3D, that is the limit of the elemental impurities into the product, which is the compliance is mainly demonstrated through risk assessment. So QRM principles as per ICH Q9 should be used for determining controls of elemental impurities. If you want to get the details of ICH Q9, we have one presentation uploaded onto the channel for ICH Q9 on how to determine the risk assessment. Risk assessment should focus on assessing the level of elemental impurities in drug product with reference to PDE values presented in the previous slide. So information for the risk assessment, that is data generated by the firm during the product development, during technology transfer, information provided by the component or excipient manufacturer and data published in the literature. Manufacturers should establish that the analytical procedure used during the risk assessment possesses characteristics, that is accuracy, precision, specificity, such that the manufacturer can be reasonably certain, that is 98% confidence level, 95% confidence level, that the measurements can be relied upon to decide control strategy. The purpose of the risk assessment is mainly 3 steps. Step 1 is to identify the potential source of elemental impurities in the drug products. Step 2 is to evaluate the presence of these elemental impurities through observed or predicted level and compare with the PDE to check and demonstrate whether they are in compliant with the required PDE levels. And third, evaluate if controls are sufficient or additional controls are needed to be implemented if the value is expected to be above the PDE. So we move on with the risk assessment step 1, that is identification of the source of elemental impurities. If you can see, various equipment processes are used for the manufacturing of drug product. So elemental impurities can be introduced from manufacturing equipment, from drug substance, from water that is used, from the container closure system due to leaching, from the excipient used, right? So continuing with the identifying the source of elemental impurity, Elemental impurities can be intentionally added at catalyst and inorganic reagent during the manufacturing process. When this is the case, they should be considered definitely for risk assessment. The identity is known and technique for controlling are easily defined because we are aware of what elemental impurities have been added. The second category can be the elemental impurities which are not intentionally added. So they are present in the drug substance or excipients and should be reflected in the risk assessment. For oral drugs, we have to evaluate the class 1 and class 2A elemental impurities. For inhalation and parenteral products, we have to evaluate class 1, class 2A and class 3 elemental impurities. They can be derived from the manufacturing equipment. This source is limited because we have the application of process knowledge, selection of the equipment, qualification and appropriate GMP. So product contact component of equipment should be evaluated for the source of elemental impurities. One assessment can be used for many potential products because same chain of equipment can be used for different products. So if we carry out the assessment for one product, it can be an overall assessment which can be used for all the products. The next category for unintentional adding can be leached from the container closure system. So it should be based on scientific knowledge and understanding of the likely interaction. When material of construction of container closure system demonstrate that it does not include the elemental impurities, so no additional risk assessment is needed. Leaching in the solid dosage form is by default minimal, so the risk assessment is not needed in this case. Risk assessment is needed in the case of liquid and semi-solid. The studies to be conducted to identify leachables from the container closure system should be in place. Risk should be assessed during evaluation of the container closure system of the product. So this table is provided in the ICH Q3D guidance, which states that what elemental impurities should be considered if they are added intentionally and unintentionally as per the route of administration. So if we see the first category, that if they are intentionally added, all the categories and all the classes of the elemental impurities have to be considered, that is class 1, class 2A, class 2B and class 3. So the risk assessment should evaluate all the type of impurities when they are intentionally added. When they are not intentionally added, so if we can see for oral drug products, we can evaluate the class 1 and class 2A elemental impurities. And for inhalation and parenteral products, we need to evaluate the class 1, class 2A and class 3 elemental impurities into the risk assessment. We move on to the next step, that is evaluate the presence of elemental impurities of the risk assessment. So evaluation during risk assessment should be based on the prior knowledge for determining the elemental impurities. It can include the data provided by the material suppliers, the data provided from the container closure system or from the manufacturing equipment. It can be the published literature that is available based on the source of elemental impurities. It can be the data generated from the similar process. It can be the testing of components of drug products by validated test methods. Testing of the drug products again by the validated test methods. The factors that can influence the elemental impurities and need to be considered during risk assessment include efficiency of removal of these elemental impurities during the further processing, natural abundance of these elements when there is a chance of their unintentional inclusion, prior knowledge of the elemental impurity concentration range from specific sources, and composition of the drug product. Variability of elemental impurity should also be considered related to variability in the analytical method, variability in specific sources, and variability in the drug product. Moving ahead, we're continuing with the step 2. The PDE reflects the total exposure to elemental impurities from a drug product in micrograms per day. Thus, PDE needs to be converted into concentration to evaluate the impurities into the drug products or drug components. This concentration concept is utilized for a drug substance and drug excipients. Separate assessment is needed for equipment and container closure system. So, If we look, there are two methodologies. We have a common permitted concentration limit for elements across the drug products. That is, the one concentration limit is given for all the components that are present in the drug products, and the permitted concentration limit of elemental impurities in the individual components. So for the first limit, it is a daily intake of less than 10 mg, then we use option 1. If the daily intake is specified or more than 10 mg, then option 2 is used. So option 2B is the permitted concentration limit of elemental impurities in individual components for the specified intake, and the option 3 is finished product analysis. So if you can see, so these two categories are divided into three options: option 1, option 2A, option 2B, and option 3. In option 1 and option 2A, a common permitted concentration limit is provided, whereas in option 2B and option 3, the permitted concentration limit is individual for all the components. We'll see in detail. The common permitted concentration limit, that is option 1, if the intake is not more than 10 grams for the drug product. It provides a simplified approach of calculation. It assumes that potential elemental impurities are present in all components of the drug products, and it calculates a common permissible target elemental impurity concentration for each drug product component. We'll see how it is done. So here is the formula for calculating the concentrations in microgram per gram for the elemental impurity, which is a very simplified formula, that is PDE in microgram per day, which we have seen into the table initially, that the defined PDE values for each elemental impurity is present. So that PDE for each elemental impurity divided by the daily amount of drug product. For option 1, the daily amount of drug product is considered common, whatever it is. Even if it is 2.5 mg, still the calculation it considers as 10 mg. So this formula is very simple for all the elemental impurities. If we see, so whatever PDE value is provided, it is just divided by 10, so it represents the value in just one point. For example, if we see the cadmium, that is CD, the PDE value for oral concentration is 5 microgram per day, which on dividing by 10 becomes 0.5 microgram per gram. So the concentration of cadmium, that is allowed concentration of cadmium, is 0.5, 0.2 for parenteral and 0.3 for inhalation. If we see, the ICH Q3 has provided an example where they have given a consideration of an oral solid dosage unit, which intake is 2.5 mg, and it consists of one drug substance and 3 excipients, for example lactose, magnesium stearate, and iron oxide. So the risk assessment identified that PB, AS, PD, and Ni are the potential elemental impurities for this drug product based on the assessment. So the maximum potential daily intake of an elemental impurity, for example, if we see for PB, that is lead, it is clearly defined as 0.5. So whatever source, it considers that all the elements, all the 4 elements, that is drug substance and 3 excipients, have by default a 0.5 concentration of the elemental impurity. Similarly is the case with arsenic. So it is 1.5. So it considers by default for all the elemental impurities present in the concentration of 1.5 microgram per gram in all the components of the drug product. Similarly with PD, the value is 10, and with nickel the value is 20. Right? So the maximum daily intake, to calculate the maximum daily intake, the concentration of each elemental impurity into the actual drug intake, in this case it is 2.5. So whatever the concentration of each elemental impurity is there, for example, if we see lead PB, it is 0.5. So 0.5 into 2.5, 1.5 for arsenic into 2.5. So it gives the value of maximum daily intake in microgram. So this value, that is 1.25, 3.75, 25 and 50, which is presented in the max daily intake, is the maximum allowed a microgram of each elemental impurity that can be present or that we can take as for the drug product. Now these obtained values are again compared with the PDEs. So we have seen in the table initially The PDE value for lead is 5, for arsenic it is 15, for PDE it is 100, and for nickel it is 200, right? So to conclude, the maximum daily dose, the maximum daily intake in microgram for lead is 1.25 and the PDE value is 5. So comparing this, the maximum daily intake in microgram is very less as compared to PDE. Thus, if these concentration in each component are not exceeded, drug product is assured of not exceeding the PDEs for each elemental impurity. So this is a very simple concept, the option 1, which gives us an overall understanding of how we need to first determine the concentration in each, it is a common concentration in microgram per gram for each of the elemental impurity. Then to determine the maximum daily intake by multiplying that concentration into the actual dose and obtain the value that is obtained, which is max daily intake, comparing it with the PDE value. So if these obtained values are less with PDE for each of the elemental impurity component, it is assured that the elemental impurities will not be present into the drug substance. Moving on with the option 2A, that is with the same formula carrying out the details for a specific dose. So it is similar to option 1. In this case, for determining the concentration, except the daily intake of assuming it as 10 mg, we just divide it by the exact dose. So permitted concentration is calculated again by the same formula. Concentration first we will determine by microgram per gram, that is PDE value divided by the amount of drug substance into the drug product. So that intake, whatever it is, we'll consider in this calculation. We'll consider the same example, like is oral solid dosage form, that is intake is 2.5 gram consisting of 1 drug substance and 3 excipients again. So risk assessment again identified the same potential elemental impurities. So maximum potential daily intake of each elemental impurity is given in this format. Lastly, we have seen for lead in the drug substance, it came out to be 0.5 because directly the PDE value of 5 was divided by considering the dose as 10, which came out to be 0.5. But in this case, if you see, for lead the PDE value is 5. Hence, as per the above equation, 5 divided by the actual dose of the drug product, that is 2.5, which gives 2 microgram per gram. Similar is the calculation for arsenic, where PDE is 15 divided by 2.5, it comes out to be 6. For PD, the value is 100. So 100 divided by 2.5 is 40 microgram per gram, and for nickel it is 200, where 200 divided by 2.5 it comes out to be 80 microgram. Now in this case also, for calculating the maximum daily dose, it is similar like we have done in the earlier case, that is the concentration of each elemental impurity into the actual drug intake, that is 2.5 gram. So the maximum daily intake which we get is 2 into 2.5 is 5, 6 into 2.5 is 15, 100 for 40 into 2.5. So now we calculate it, now we compare these values with the PDEs. So in this case also, no elemental impurities should exceed their PDE. Thus, if concentration of each component are not exceeded, drug product is assured of not exceeding PDEs for each elemental impurity. Components may be used in any concentration in the drug product. So this approach is very much similar to the approach that is given in option 1, except the case that in option 1, irrespective of the dose, the PDE value is divided by 10 to get the concentration, where in this case the PDE value is divided by the actual drug intake, that is the specified amount. Now we will understand the option 2B. Here the permitted concentration limit for each elemental impurity is defined separately. In the previous two examples, that is option 1 and option 2A, the elemental impurities, for each elemental impurity there was a common defined limit for each of the elemental impurity. For example, we have seen in lead they gave in option 1 a limit of 0.5 and in option 2 for whatever components it is. But in this case we will see somewhat different. So the firm may set permitted concentration of each element based on distribution of elemental impurities in the component. This allows the maximum permitted concentration of an element in some component may be higher than option 1 or 2A. However, this should be compensated by lower allowable concentration in other components. Additional knowledge on content of each elemental impurity in component of drug product is needed. For each elemental impurity, maximum expected mass of each elemental impurity in the final product is calculated by multiplying the mass of each component material into the permitted concentration established in each material and summing all the components. So here it seems to be quite a complex formula, like PDE in microgram per day should be greater than or equal to the sum of CK, where CK is the permitted concentration of elemental impurity in the component and MK is the mass of the component in the maximum daily intake of the drug product. So we'll see with the example of the same example, that is oral solid dosage form of 2.5 gram intake consisting of one drug substance and 3 excipients. Again, same elements have been identified in the risk assessment. In this case, additional knowledge on the component of each elemental impurity is needed for the drug product. So the data for concentration of elemental impurity in the components and mass of each component in the product, these two important things are needed. If we see, for example, the data of concentration of elemental impurity in the component, this is the additional knowledge that we need during the evaluation through option 2B. So if we see, for example, lead, it is below LOQ for drug substance, 0.5 for arsenic, 20 for PD and 50 for Ni. And lactose, it can vary, that is it can be 0.1 in magnesium stearate, it can be 0.5 in iron oxide, it can be 10. So in the option 1 and option 2A, we have seen, for example, the lead PB value, it was considered same for all the components. In this case, if you see, the values are different and it is based on additional knowledge data from the input of different sources. So if we see the drug substance, it can be below LOQ, lactose can be 0.1, magnesium stearate can be 0.5. So there are different limits that can be defined. And mass of each component in the product. So this is also very important. How much is the quantity of that specific drug product or specific component into the drug product? For example, 0.3 gram of drug substance, 0.2 gram of lactose, 0.4 gram of magnesium stearate, or 0.1 gram of iron oxide, etc. This is just an arbitrary example. So when we consider this calculation, that is by multiplying the data for concentration of elemental impurities into the mass, and on obtaining of this value, we sum all the values. For example, for lead, we calculate the sum of all the potential elemental impurities into mass, and we sum all the data to get a consolidated value for lead. Using this equation, level of elemental impurity in the product can be determined, and it is again compared with the PDE value. So it should not exceed the PDE value that is provided into the table. And finally, option 3, that is the finished product analysis part. So concentration of each elemental impurity can be measured into the finished product. Maximum concentration of each elemental impurity may be calculated using daily intake of product and PDE of that elemental impurity. The total mass of elemental impurity should not be more than the PDEs. If we see the formula, it is again the same formula for this drug product, that is PDE in microgram per day upon the daily amount of drug product in gram per day. For example, for option 3, that is finished product analysis, if we consider the same example of daily intake of 2.5 gram consisting of the same excipients and the same elemental impurities, if we see, the values that are obtained here is similar to the values that are obtained in option 2A, because the PDE value gets divided by the amount of drug substance, that is 2.5 mg. So it is a common limit for all, and the values obtained is 5, 15, 100, which is the elemental impurity concentration into the amount of drug, that is, for example, for lead 2 into 2.5, because 2.5 is the concentration of the drug, and then it is compared with the PDE. So total conclusion is the total mass of each elemental impurity should not be more than the PDE, right? Step 2, again continuing the step 2, conclusion of the risk assessment. Now we have seen and evaluated the elemental impurity either by using option 1 option 2A, option 2B, or option 3. Now we'll conclude the risk assessment, and the conclusion should be well documented along with all the methodology that has been adopted. So the conclusion can be there are no potential elemental impurities into the drug product. There are one or more potential elemental impurities into the drug product, and it should also conclude if there is one source of potential impurity or multiple sources of potential elemental impurity. It should also be noted that observed or predicted elemental impurities significance should be evaluated against the defined PDE value. So the PDE value and the observed level or the potential level of elemental impurity should be compared. The control threshold as defined by ICH Q3D is 30% of the PDE value for an elemental impurity into the drug product. So we have a control limit. It should be not more than 30% of the PDE value for any component for any elemental impurity, right? If the total elemental impurity level from all sources is below 30% of the PDE for whatever sources, for example, we have seen the excipients and drug substance example in option 1. So if all the component, including all the component, the PDE value, the obtained value of elemental impurity is less than 30% into the drug product, then no additional control measures are needed. However, if the risk assessment conclude that the obtained value is more than 30% in all the cases, then controls should be established. Which takes us to the step 3 of risk assessment, that is control of elemental impurities. So how we are going to control? If step 2, the control threshold of 30% PDE value is exceeding, the firm need to implement additional control measures, which can include modification of the manufacturing step to include specific or non-specific purification process, implementation of in-process of upstream controls, establishment of specification limits for excipients or materials, establishment of specification limits for drug substance or drug product, selection of appropriate container closure system. Periodic testing may be applied to elemental impurities for identification. The USP Chapter 233 defines 2 testing methods. The procedure 1, it gives ICP-AES or atomic absorption spectroscopy, and the procedure 2, that is ICP-MS, that is mass spectroscopy. It also tells that alternative methods can be used. Each method should be appropriately validated. Also, the guidance does not require routine testing of all the drug products for elemental impurities. Alternative approaches for control, as discussed above in the yellow color, are used and are acceptable depending on the risk assessment. For example, the routine testing of APIs and excipients. If potential risk of introduction of elemental impurity in product is low, it is not needed to test that elemental impurity in each lot. If changes to the drug product or drug component has potential to change the elemental impurity content, the risk assessment and established control should be reevaluated. Such changes includes changes in the synthesis route, excipient supplier, raw materials, equipments, and container closures. And continuing with the step 3, for submission to the regulatory authority, variability for the elemental impurity data can be established by providing data of 3 representative production scale lots or 6 representative pilot scale lots. The submission document should include identification of the elemental impurities, their sources, their acceptance criteria, and their control methodology. The information related to speciation of elemental impurities, that is their oxidation state, is not always needed and not to be provided to the regulatory authority. When total elemental impurity level in component used in risk assessment, the information related to release of elemental impurity from component in which it is found is not needed. This brings us to the end of this presentation. Hope you have liked this presentation. Kindly subscribe to Prof Talks for more educative videos. Thank you.